in nature, and this group of bacteria, it has played very important role for the development of life in the world. Before starting this study of this bacteria, firstly we are going to study one of the most important differentiation characters between the prokaryote cells and the eukaryote cells. These prokaryote cells, they includes bacteria, while the eukaryotic cells, they include animals, algae, fungi, protozoa and plants. Now you also see here there is a variation in size of these prokaryotic cells and the eukaryotic cells. The prokaryotic cells, they are generally measured in 1:2 into 1:4 micrometer in length or size. While the eukaryotic cells, they are having diameter more than 5 micrometer in diameter or width. Now in case of prokaryotic cell, there is a presence of a nuclear material is there in the form of nucleoid or chromatin bodies. But in case of eukaryotic cell, this nuclear material, it is mostly located in chloroplast, mitochondria and the nucleus. But in case of prokaryotic cell, this nuclear material, which we call it as a nucleoid, it is particularly located in the cytoplasm of that eukaryotic cell. When we observe the structure of this nucleus, in case of prokaryotic cell, there is a presence of only one circular chromosome is there. While in eukaryotic cell, there is a presence of more than one chromosome. In prokaryotic cell, this one chromosome, it is not having any boundary around itself, means it is not surrounded by the nuclear membrane. But in case of eukaryotic cell, these chromosomes, they have been surrounded by a nuclear membrane. So because of this character, that whether the nucleus is well developed or not, depending upon this character, we have differentiated the cells into two types, prokaryotic cells and the eukaryotic cells. These prokaryotic cells, they are not having well developed nucleus, while the eukaryotic cells, they are having well developed nucleus. When we observe the chromosome of prokaryotic cell, there is an absence of histone protein. There is an absence of histone protein. While in case of eukaryotic chromosome, you can see there is a presence of histone proteins, and around that histone protein, this chromatin network get coiled in order to form the chromosome. Now in case of prokaryotic cell, there is no mitotic division is there. But in case of eukaryote, there is a mitotic nuclear division is there. In case of prokaryotic cell, there is absence of nucleolus is there. In case of eukaryotic cell, there is a presence of nucleolus is there. In case of prokaryotic cells, the genes which are related for a particular functions, they are clustered. And the genes which are clustered in case of prokaryotes, they are called as operons. In case of eukaryote, there is no clustering of functionally related genes, means they are far apart from one another. In case of prokaryotic cells, the zygote is partially diploid, while in the case of eukaryotic cell, zygote is diploid one. Now, when we observe the cytoplasm of this eukaryotic cell as well as the prokaryotic cells, you are able to clearly differentiate that in case of eukaryotic cells, different membrane bound organelles are there. Now, in case of prokaryotic cells, cytoplasmic streaming, it is absent while it is present in eukaryotic cell. Pinocytosis, it is absent in prokaryotic cell, but it is present in eukaryotic cell. You may observe presence of gas vacuoles in eukaryotic cell. And the gas vacuoles, they are present in they are absent in eukaryotic cell, but they are present in prokaryotic cells. Mesosome, which is the major structure which is present primarily in prokaryotic cell, but it is absent in the eukaryotic cell. When we observe the protein synthesis machinery, in case of prokaryotic cell, it is 70S type, and in case of eukaryotic cell, it is 80S type, which is primarily bound to the endoplasmic reticulum. While in case of mitochondria and chloroplast, which also contains the chromosomal material, there is a presence of 70S type of ribosome. In case of prokaryote, there is absence of mitochondria, but in case of eukaryote, it is present there. Chloroplast, it is absent in prokaryotic cell, but it is present in eukaryotic cell, in particularly to the algae and the algae and the plants, where this chloroplast it is present. In some protozoas also it is present, but it is absent in fungi and animals. Golgi bodies, they are absent in prokaryotic cells, but they are present in eukaryotic cells. Endoplasmic reticulum, it is absent in prokaryotic cell, but it is present in eukaryotic cell. Membrane bound vacuoles, they are absent in case of prokaryotic cell, but it is present in eukaryotic cells. So this is how you can differentiate the eukaryotic cell from the prokaryotic cells. And the major examples of the prokaryotic cell is the bacteria, and the eukaryotic cells is the animals, plants, algae, fungi, and protozoa. So this is about the differentiation of the prokaryotic and eukaryotic cell. Now we begin our today's another point, that is the general characters of Archaebacteria. We have seen the differentiation between the cells. We have differentiated the cells into two types, eukaryotes and prokaryotes. In case of prokaryotes, there is a presence of bacteria. Now in case of bacteria, there is also two groups are there, eubacteria and Archaebacteria. These are the two groups of bacteria which are present there in nature. True bacteria, we call it as a eubacteria. These are the present time bacteria are there. While these Archaebacteria, they are oldest bacteria which are present in the environment or in the nature. This Archaebacteria, this word, it is derived from Greek word archaeos, means ancient. This bacteria, Archaebacteria, it is called as ancient bacteria. because it is the oldest bacteria which is present in the environment. Now, by carrying out different experimental experiments, by using the study of ribosomal DNA, this ribosomal DNA it is extracted from these bacteria, and the studies is carried out of these two group of bacteria, Archaebacteria and Eubacteria. From these studies of ribosomal RNA, it realized that these two group of bacteria, they are separated very early stage in the evolution of life on this planet. Means these two bacteria, they are the separate group of bacteria which are derived from one bacteria, but they are separated very early stages in the evolution of life on this planet Earth. So these are the two bacteria which are totally different now, but at the early stages they are having same development stage. Now you can easily differentiate between these bacteria, Archaebacteria and Eubacteria. There is a difference in the cell wall of the bacteria. Now in case of Archaebacteria, the peptidoglycan, which is the major component of cell wall of Eubacteria, it is totally absent in case of Archaebacteria. In case of cytoplasmic membrane of Eubacteria, there is a presence of long chain fatty acids which are bound together by glycerol, which are bound to the glycerol by ester linkage. It is present in Eubacteria, but it is absent in Archaebacteria. The plasma membrane of Archaebacteria, it contains long chained branched alcohols bound to the glycerol by a ethyl linkage, which is present in Archaebacteria, but it is absent in Eubacteria. When we observe the protein synthesis characters of these two bacteria, it is found that in case of Archaebacteria, there is an involvement of methionine, which is the first amino acid which is to be added during the protein synthesis. It is added in the Archaebacteria. But in case of Eubacteria, N-formyl methionine, this is the first amino acid which is to be added during the protein synthesis. Now in case of translation process, the translation process in Archaebacteria, it can be easily stopped by using diphtheria toxin. When we use diphtheria toxin, it stops translation process in Archaebacteria, but it is not affecting the translation process in case of Eubacteria. Chloramphenicol, it is the antibiotic which is affecting the protein synthesis in case of Eubacteria, but it is not having any effect on the protein synthesis, on the translation process in case of Archaeobacteria. So this is how these two bacteria, they have been different. They are different from one another in terms of their cell wall, in terms of their protein synthesis process, in terms of their plasma membrane structure. Now, in this class, we are going to study the general characteristics of this Archaebacteria. This Archaebacteria, it is not a perfect homogeneous group. But in case of Eubacteria, you can observe there is a heterogeneous groups are there. Various types of bacteria are there which are having various characters. And these two groups of the bacteria, they are differentiated from one another by their morphology, by their metabolism, by their chemical composition, and by their habitat. These archaeobacteria, they are most commonly found in some places only. While these eubacteria, they are commonly found everywhere. These archaebacteria, they are limited to some places only. And these archaebacteria, which are found in the nature, they are divided into three different types of group. First is the methanogenic bacteria. Second is the extreme halophiles. Third one is the thermoacidophiles. And these three bacteria, they form the Archaebacteria group. These are the three major Archaebacteria which are present in the nature, and they contributing to the nature in a different manner. Now, one by one, we will see the characters of this group of Archaebacteria. First is the methanogenic bacteria. Why the name methanogenic bacteria is given to this bacteria? It is because the end product of this growth of this bacteria, it is a production of methane gas. This is the end product of growth of this methanogenic bacteria. There is a production of methane gas and water. These methanogenic bacteria, they are hardcore anaerobes. Means they require absence of oxygen condition to their growth. When they are exposed to the oxygen, they will immediately die. So these bacteria, these methanogenic bacteria, they are hardcore anaerobes. They only grow in the absence of oxygen. Now here, when we see the photosynthetic plants, they use sunlight as a source of electron for their growth, for the photosynthesis process. But in case of methanogenic bacteria, they use hydrogen or formic acid as a source of energy, as a source of electron. And carbon dioxide as a source of carbon to form the methane gas. So these methanogenic bacteria, they are autotrophs. They can obtain their own energy, and they can obtain their own food by using this process, where there is a use of hydrogen gas and carbon dioxide gas. These two get combined. Here hydrogen gas, it is acting as a source of energy, electron, and while the carbon dioxide, it is acting as a source of carbon, and this is utilized by this methanogenic bacteria to grow. And during their growth, they form the methane gas and water molecule. But there are some of the methanogenic bacteria which also requires another components for their growth. When we try to grow these bacteria in our lab, we have to supply this bacteria a complex medium. That complex medium must contain different types of minerals, salts. So we can grow this bacteria, methanogenic bacteria, in our lab by using complex medium, but we have to provide also the anaerobic condition to this group of bacteria, so they will grow there. Otherwise, when we try to grow this bacteria in aerobic conditions, these bacteria won't grow there. They will not grow there. So which are the common habitats where these methanogenic bacteria, they are found? These methanogenic bacteria, they are found whenever there is a presence of anaerobic habitat is there. First requirement for the growth of this bacteria, it is the anaerobic habitat. Second requirement, that habitat must be rich in organic matter. And this organic matter and the anaerobic habitat, these are the two essential conditions which are required for the growth of this bacteria. This non-methanogenic bacteria, they utilize this organic food to generate hydrogen and the carbon dioxide gas. So the most common habitats where this methanogenic bacteria they are present, they are the marine sediments, swamps, marshes, ponds, lake mud, intestinal tract of humans and animals, rumen of the cattle. These bacteria they are also observed in anaerobic sludge digesters, where we carry out the sewage treatment before releasing the water into the river. So these are the locations where these methanogenic bacteria they are commonly found. When the study of this bacteria is carried out, it is found that there is a presence of two uncommon enzymes are there, which are only present in the methanogenic bacteria. It is not observed in any other microorganisms. It is particularly present in this methanogenic bacteria only. Which are these enzymes? First is the coenzyme M. This coenzyme M, it is directly involved in methyl transfer reaction. Second one, coenzyme F420. It is a flavin-like chemical compound which is present in this bacteria in order to carry out anaerobic electron transport system. Means this coenzyme F420, it is particularly present in electron transport of anaerobic bacterial cell. And now this coenzyme, it is also having another character. When this coenzyme it is exposed to UV light, it fluoresces. It shows bright light. So, in order to detect the presence of this bacteria, you can use this technique. Expose these bacteria to UV light. If these bacteria, methanogenic bacteria, they are showing the fluorescence, we can say that this bacteria it is belonging to the methanogenic bacterial group. Now, when the classification of this methanogenic bacteria is carried out, this classification it is majorly based. It is majorly based on their morphology and their Gram reaction. Here, cell wall composition of this bacteria they are also taken in consideration while classifying this methanogenic bacteria. So, this methanogenic bacteria they are divided into 7 different genus. First genus is the Methanobacterium, which is showing gram positive to gram negative reactions, and these are the long rods. They are non-motile, and when the cell wall of this bacteria they have been studied, it is made up of pseudomurine. Second genus is the Methanobacterium. It is a gram positive, lancet shaped short rods or cocci. They are non-motile. They are also having pseudomurine in their cell wall. Third genus is Methanococcus, which is having gram negative pleomorphic cocci. They are motile. They are having one flagellar tuft at the end. And the cell wall of this bacteria is composed of protein which is having some amount of glucosamine. Fourth genus, it is a methanogenum, which is also having gram-negative pleomorphic cocci. They are motile by peritracheous flagella, and the cell wall it is totally composed of protein component. Fifth genus, it is a methanomicrobium. These are gram-negative short rods. They are motile by single polar flagella. And the cell wall of this methanomicrobium, it is made up of protein components, not the peptidoglycan, which is observed in eubacteria. This pseudomurine, it look like the peptidoglycan, but it is not the peptidoglycan. It is chemically different from the peptidoglycan component, which is present in the cell wall of eubacteria. Then the sixth genus is the Methanosarcina. These are the gram-positive cocci which are present in cluster. Motility is not there. And the cell wall, it is made up of heteropolysaccharide. Then the last genus, it is a Methanospirillum. This is the gram-negative long, wavy, filamentous or curved rod. And they are having polar flagellas there. And the cellular composition of this group, this genus, it is not studied now. You can see these are the methanogenic bacteria which are present there in the nature, and these are particularly found in such places where there is a anaerobic condition it is developed. Marshes, ponds, sewage digesters, intestinal tracts of human beings as well as animals, in the rumens of the cattle. These are the favorite places where these methanogenic bacteria they grow very well. Then second group of bacteria, they are the Archaeobacteria. In the Archaeobacteria, it is the extreme halophiles. Halophiles, the name it is this halophiles is given to this bacteria because halo means salt, philes means living. Halophiles are those group of bacteria which are loving the salts, because in the presence of high salt concentration these bacteria only grow. When we are not providing high salt concentration to this bacteria will not grow in the lab. This extreme halophilic bacteria, they are aerobic organisms, means they require oxygen for their growth. They are chemoorganotropic bacteria which require 17 to 23% of the sodium chloride concentration for their normal and good growth. See here, this much concentration is required by this bacteria to grow. Now in such a high salt concentration, not other bacteria can be growing there. Only halophiles, the extreme halophile, these Archaebacteria only grow in such a high salt concentration. This extreme halophile bacteria, they are gram-negative bacteria. In case of halophilic bacteria, you are able to observe variation in the shapes. You may observe rod-shaped or disc-shaped bacteria here. You may also observe spherical to ovoid-shaped bacteria in case of extreme halophilic bacteria. And these halophilic bacteria, they are most commonly found in salt lakes. For example, Great Salt Lake, the Dead Sea. Also, these bacteria, they are most commonly present in industrial plants which are generating the salt by solar evaporation of the seawater. This bacteria is also present in fishes which are preserved by using large amount of sodium chloride salts. That is salted fishes. Here in this habitat, this extreme halophilic bacteria, they are prominently growing there. Now when the cell wall of this bacteria is observed or studied, then there is a difference in the cell wall of this halophilic bacteria and the U bacteria. These halophilic bacteria, they are having pseudomurine as their cell wall component, which is different from the eubacterial peptidoglycan component. Now in case of eubacteria, there is a presence of N-acetylmuramic acid in the peptidoglycan layer. But in case of extreme halophilic bacteria, there is a presence of N-acetyloaminouronic acid. This is the component which is present in the cell wall of extreme halophilic bacteria. Then there is a presence of in the cell wall of the pseudomurine of that is a halophilic bacteria. There is a presence of tetrapeptide L-amino acid chain. And this L-amino acid chain, it is having at the terminal position glutamic acid. So this is the difference between the eubacterial peptidoglycan and extreme halophiles pseudomurine. You can see here, this is the halophilic bacteria. You can observe there is a halococcus is there. This is a halobacterium, and these are the places where these bacteria, they are present in tremendous amount. Whenever there is a presence of salt in higher concentration, at these places you are observing this group of bacteria. Now when we grow this bacteria on medium, you can observe different kinds of coloration which are shown by the colonies of this bacteria. You can observe red to orange coloration which is shown by this group of bacteria when we grow this bacteria on the medium. And this coloration which is shown by this bacteria, it is because of the presence of a pigment, carotenoid pigment, which is present in this group of bacteria. And this carotenoid pigment which is present in this group of bacteria, it gives protection to this bacteria from the effect of sunlight, and in the sunlight there is a presence of UV light. So because of the presence of this carotenoid pigment in this group of bacteria, it gets protected from the effect of UV light which is present in the sunlight. Now there are various features which are shown by this group of bacteria. This halophilic bacteria, they resist dehydration, particularly at high sodium chloride concentration. by maintaining inside their cell higher concentration of potassium chloride. Inside this halophilic bacteria, there is a presence of high concentration of potassium chloride, while outside the bacterial cell, there is a presence of high concentration of sodium chloride. So, by maintaining this high concentration of potassium chloride, it resists the dehydration which is caused by because of this high sodium chloride concentration. Now, in case of this halophilic bacteria, the ribosomes and the cytoplasmic membranes, they are only stable when there is a presence of high concentration of potassium chloride. Similarly, the enzymes which are functioning in this bacteria, they only functions in this bacteria when high salt concentration of sodium chloride and or potassium chloride it is present in this bacteria. So dehydration it is resisted by this bacteria by maintaining this potassium chloride concentration. The ribosomes and the cytoplasmic membrane it only present there when the concentration of potassium chloride it is high. Similarly, the enzymes which are there in this bacteria, they only functions when there is a presence of high concentration of sodium chloride or potassium chloride. Otherwise, these enzymes will not function. Now, in case of halobacterium, the cell wall of this halobacterium it is made up of certain protein subunits which are held together in the presence of sodium chloride only. If the concentration of sodium chloride it falls below the certain level, approximately 10% weight by volume, these protein components they get separated, they get break up. When the study of halococcus bacterial cell wall it is carried out, it is majorly made up of complex heteropolysaccharides. And this cell wall of this halococcus bacteria it is stable at lower concentration of sodium chloride. At lower concentration of sodium chloride. Now, in order to generate ATP, these extreme halophiles they carry out different types of processes to obtain the energy. These halobacteria they are aerobic in nature, so they use the electron transport system to generate the proton motive force to obtain the ATP synthesis. Now these halobacteria they also obtain the energy by fermentation of arginine amino acid. And when this bacteria, halobacteria, ferments the arginine amino acid, it ferments this amino acid in anaerobic condition. So this bacteria can grow as an aerobic bacteria as well as anaerobic bacteria. And when it is growing in anaerobic condition, it ferments the arginine and obtains the ATP. So ATP which is required for the cells to function. Third source of ATP generation is that in this halophilic bacteria, there is a presence of a purple pigment is there. And that purple pigment, it is called as bacteriorhodopsin. And this purple pigment, It is produced by this bacterial it is mainly present in the bacterial cell membrane when low oxygen level is there. And when this bacterial rhodopsin pigment it is exposed to the UV light, it shows bleaching or it shows fluorescence. And this bleaching phenomena it is used to generate the proton motive force, and which results in the synthesis of ATP molecules in this bacteria. So this bacteria it can obtain energy by using aerobic electron transport system, which is generally present in aerobic organisms. Secondly, they obtain the energy by fermentation of arginine. Thirdly, they obtain the energy ATP in the form of ATP by using the bacteriorhodopsin pigment, which is present in the cell membrane of this bacterial cell. Now, third group of Archaebacteria, they are called as thermoacidophiles. As the name suggests, they require acidic conditions for their growth. They require high temperature condition for their growth. These thermoacidophilic bacteria, they are aerobic, gram-negative bacteria, and which requires extremely high acidic pH for their growth and considerably elevated temperature for their growth. So these two conditions must be satisfied in order to grow this thermoacidophilic bacteria. Now these thermoacidophilic bacteria, they are divided into two prominent genera, Thermoplasma and Sulfolobus. These are two groups which are present in thermoacidophiles Archaebacteria. Now first we see about these thermoplasmas. These thermoplasmas, they are chemoorganotropic microorganisms. When we observe the structure of this thermoplasma, they similarly look like that of a mycoplasma. This mycoplasma, it is a group of organism which doesn't have any cell wall. So because of the absence of the cell wall, this mycoplasma, it shows pleomorphism. Any kind of shape can be shown by this group of bacteria. So you may observe spherical shape in case of thermoplasma. You may observe filamentous shape in case of thermoplasma. So there is a variation. Pleomorphism is there. And the ideal temperature which is required for the growth of this group of bacteria, it is in between 55 to 59 degrees Celsius temperature. Minimum temperature which is required, it is 44, and the maximum temperature which is required here, it is the 62 degrees Celsius temperature. But the optimum pH which is required by this group of bacteria, it is 2 pH. When we try to grow this bacteria in neutral pH, this bacteria undergoes lysis process. Means they can go autolysis at neutral pH. These thermoplasmas, it can be easily isolated from the burning coal refuse which is used in generating the electricity. Which is obtained from the coal burning process. So this is the places where this thermoplasma bacteria can be easily observed. It is easily growing there. You can see here, this is the electron microscopic structure of the thermoplasma bacteria. It can taking any kind of cells there, shapes there. Then second group of thermoacidophilic bacteria, it is the sulfolobus. As the name suggests that it shows lobe-shaped or pedicle shape in appearance. They have a definite cell wall which is majorly made up of only the protein component. This sulfolobus bacteria, it can grow in an optimum temperature range between 70 to 87 degrees Celsius temperature. And at such high temperature, any other eukaryotic bacteria, they are not able to grow there. Bacillus, it is not growing there. E. coli, it is not growing. So this is a special character which is shown by this sulfolobus bacteria. And the optimum pH which is required for the growth of the sulfolobus bacteria, it is optimum pH is the 2. This is the essential pH which is required by this for the growth of this bacteria. And this sulfolobus bacteria, they obtain the energy by autotrophic process, which is a facultative autotrophic process, means they can use two different mechanisms to grow. First mechanism is the chemolithotrophs, where sulfur element it is used as an electron donor. Second is the chemoorganotrophs. Here, this chemoorganotrophs, it contains the organic substrates. And this sulfolobus bacteria, it naturally present in sulfur hot springs around the world. In our Nanded district, there is a process of Unkeshwar, where there is a hot spring, sulfur ponds are there, where this bacteria can easily prominently grow there, because there is a sulfur content is there in that pond, as well as there is a high temperature is there, low pH is there. So this is the environment which is suitable for the growth of this sulfolobus bacteria. And this low pH bacteria, they can also be seen in various places in the world. So you can see this is the place where this sulfolobus or thermoplasma bacteria, it can be easily growing there. And this is the bacterial cell, sulfolobus bacterial cell, which is observed under the electron